There's been a couple of bits of news about ongoing HIV gene therapy trials, which I figured I could just share while everybody's here. And then, the information on the rescheduled webinar with Dr. Hauber will be sent when we're able to confirm that date with him. And so I'll just share my screen and put up it's only 3 slides.
All right.
So Richard, you want questions as you go or at the end, or what's your preference?
Either is fine.
Okay. So jump in, anybody. We've got plenty of time today.
So the first bit of news is about the topic of I think the webinar, not the last one, but the one before, which is using this gene editing tool called CRISPR-Cas9 to try to, it's sort of like a genetic surgery that cuts the genetic code of the virus out of cells that is infected. And so this trial has been just got started quite recently. And on September 15th, the manufacturer, Excision Biotherapeutics, put out a press release just to announce that the first person has been dosed with this gene editing tool. It's delivered into the body with a sort of harmless virus vector, generally harmless virus vector called adeno-associated virus, or AAV. And the first participant has received it. They haven't exploded. Thankfully, nothing immediately untoward has happened. So that's good news. But obviously, there's concern about potentially long-term effects. And so, as is required for all gene therapy studies in the US, there'll be long-term follow-up out to 2037 for people, or the small number of people in this trial. And the way the trial is designed is that they'll be doing interruptions of antiretroviral therapy after 12 weeks after the administration. So if everything is still okay, probably that first participant will be maybe beginning to initiate an ATI sometime around now. And then they'll get some, be able to look at whether the approach has reduced the amount of viable HIV enough to delay or rebound of virus, or lead to better control of viral load after antiretroviral therapy is interrupted. So that's kind of a key milestone for this trial, but a lot more to learn. And that's just
Richard,
a bit.
Do you remember, do they get more than one dose, or are they just dosed one time with the?
It's a single dose study, I believe. Okay. I will double check that.
Okay.
And then the other bit of news, if I can persuade the slides to advance, is that about a slightly different gene therapy approach. The company pursuing this is called American Gene Technologies. They're a little bit prone to quite sort of hypey press releases about the approach, hoping that it may be curative. It's still a long way from knowing whether that is possible. It's a little different. We've had other, Sangamo is the company most famous for doing an approach that was based on trying to genetically modify people's CD4 T cells to make those cells resistant to the virus. And there were some signs of that approach could work, but not enough for the company to pursue it. And what was interesting is that in the people that seemed to get the best results, it seemed that the approach seemed to have particularly boosted their T cell responses to HIV itself. And so this company decided that they would really try and focus protecting not all CD4 T cells or just general CD4 T cells, but the CD4 T cells that are specifically targeting HIV. So they sample from participants, expand the CD4 T cells that are targeting the virus in the laboratory, modify them with this gene therapy that's designed to make them resistant to the virus, and then reinfuse them in large numbers back into the participants. And the phase 1 trial has been underway for a while. There's been press releases reporting that so far the intervention has been safe, and the Data Safety Monitoring Board, which reviews trials independently, has allowed additional the dosing of additional participants to proceed, and has now given permission for a follow-on study which will involve an analytical treatment interruption. So this is really the big test for this approach, is whether these CD4 T cells targeting HIV that are now hopefully protected from infection can actually do a better job of controlling and sort of leading the immune response to HIV itself, and maybe control the virus when antiretroviral therapy is stopped. There's some people have already started the ATI. There was a press release from the company announcing that this had started, and they're hoping they might have results to share by the end of this year. And really, that's, although there's been, the company's been very, eager to hope that this is going to work, bullish about it. Really, if it, this is the ultimate test, and if there's no evidence that these cells are able to mediate better control of HIV viral load, then the prospects for this approach are not going to look so good. Again, like with EBT, the safety monitoring is long term, going out to 2038.
That's one of the things I would love to discuss with other people on this call, is about this safety monitoring for 15 years. Because we've already seen that the first company to do this for HIV sort of folded its HIV wing, and people from those studies have not been followed. Not in the way that we like to think when they say safety monitoring for 15 years. And I don't know what people think about that. There doesn't seem to be any sort of backup to make sure that people are followed. There's no sort of protocol that I know of, that whether it is the company doing it or someone who takes over for the company, the sorts of things that we would want them to follow. I don't know if there's been any discussions for gene therapy participants. And I don't know if you're aware of anything like that, Richard. But
Sure. Yeah, I'm not sure exactly how it works, but there is a long-term follow-up study for participants in Sangamo trials.
Okay.
There seemed to be a delay before it was initiated, and so it seems, I think maybe they lost some people because of that. And I'm not sure exactly how it's being administered. We should maybe look into that. It would be an interesting kind of topic to do maybe at some point, just to find out how that all works from FDA and so on, because in addition, there's a trial registered in clinicaltrials.gov for that follow-up. And there was another company that was created by the quite famous scientist called David Baltimore that studied a gene therapy called CAL1. And they also have a long-term, that company has since gone under and got bought by somebody, I think, or some of the technology got bought, but somehow that long-term follow-up study is still happening for people who received that HIV gene therapy. It's also in clinicaltrials.gov. So there must be some kind of mechanism to make that happen. I'm just not sure exactly how it works.
I think they're not like you would, like you're still on a protocol. They're often, you check in. It's kind of like the CDC and your vaccine stuff. I mean, if anybody's enrolled in their program, that VAERS program, to check up on you, it's very different than what you consider a protocol follow-up. And I know, Michael, that we both know somebody that has had wonderful results, and we can't get anybody to write about it or whatever, because he wasn't followed per se. And I think that was really on his doctor, but that's a whole nother story. But interestingly, I got in touch with the FDA a good 3 or 4 months ago now and asked them, we've done every year I do a thing with CEDAR, the drug side of the FDA, and they give us an update and whatever. And I try to get them, CBER, the biologic side, to do a call with us so that we could talk about all of this sort of stuff. But, they never got back to me, and I really did, rattle a lot of cages. But maybe next year I will, continue to. Well, I'll continue to rattle cages, and maybe next year we can figure out something, because it would be great to know. And the next time we have a call with Excision or any of the companies, it would be great to know what exactly that follow-up looks like.
Right.
So, and what we think it should look like. I think we have some people in
Yes.
some hands up. Michael, you
Yeah,
want to
Alistair.
take that?
Alistair and then Jeff. So Alistair, why don't you go first?
Can
I
we
was just
announce
starting.
first, though? Wait a minute, Alistair.
Sure.
Sorry, honey. That Dr. Halbert was unable to join us today. He is out of the country, out of his country, and his computer blew up, and he just can't get it to work. So we're going to reschedule him and use this this time to discuss different issues. We're on safety now and long-term follow-up. Go ahead, Alistair. I'm sorry.
Not a problem. It saves me a whole bunch of typing. I was one of the first 3 participants in the first Sangamo autologous stem cell gene modification study, where they took adult stem cells out of me, modified them, put them back into me again. And that ended up with me being followed for a full 20 years until several years ago, when they basically said enough is enough, nothing has happened, no new heads have grown, nothing has fallen off that shouldn't. At this point, we are no longer going to be taking the regular blood tests that we're taking, and just checking up to see if anything stranger than usual had happened with me. So as far as I know, the 20-year was or is a standard. The first study that Dr. Jefferys talked about appeared to be a 15-year follow-up rather than a 20. So they may have actually eased it because of what they found or didn't find during the 20-year follow-ups. But as far as I know, from my experience, it was a 20-year follow-up.
Yeah, and the company can also, you know, I guess petition is not the right word, but request that the follow-up time be shortened because of the kind of things that you've talked about, no heads growing or whatever. But Alistair, what kind of follow-up did you have to go through? How many time, you know, can you share that with us?
For the first, I don't remember, it was 5 or 10 years. It was several times a year. And then it went to twice a year, and then it went to once a year. And I don't know, I don't know when Sangamo actually went out of business or went out of this particular business. And as such, I can't speak to whether it was done by the care clinic and Dr. Mitsuyasu, who was the principal investigator at UCLA, or if it was done because as part of the setting up of the study, funds had to be allocated to each of the investigative sites so that they would be able to and would follow through on following up on people for the full 20 years.
But so what did you just have a blood draw when you went in, vitals taken and that sort of thing?
For the first few years, it was blood draws, vitals taken, questions about anything that I'd noticed, anything that they had noticed. Since I have taken part in a fair number of studies through the UCLA Care Clinic, to me it was just another study basically that I had great hopes for, because to me that made a lot of sense. That if you could arrange it such that a patient's own stem cells could be put back in a form that would actually settle into the bone marrow and replicate from that point on as the dominant strain, that would be, to me, much more ideal than having to go through a full immune system wipeout and then replacement.
Right. Plus, we know somebody who hasn't been on therapy since. So, you know, and nobody know, you know, there's not an official record of that. So it's,
right.
We also know somebody that did very well for a really long time afterwards. So it's just it's a shame that all that wasn't captured.
Right.
The way Dr. Mitsuyasu explained it to me was that they learned from the first 3 that they needed to take out more of the adult stem cells from each participant. And also they needed to multiply them outside the body so that much higher numbers were put back in. But they were able to detect the cells during endoscopies in the early years, not endoscopies, flexible sigmoidoscopies in the early years. They were able to detect the cells in decreasing amounts over that first 5 years, even with the comparatively small amount that they put back into me.
Oh, that is interesting.
Can we maybe give another quick update on Dr. Halber, just
Yeah.
because a bunch of
Sure.
sort of people.
Yeah, I'm sorry that I keep repeating this, but we keep having new people join. Unfortunately, Dr. Halber contacted us last night. He is from Germany, but in Denmark, his computer just would not function. I mean, he had a number of people try to help him work it out. But so we're just going to talk about issues of interest during this session. We just didn't want to waste the time, and we're going to try and reschedule him. I actually emailed him last night with another date, but I haven't heard back yet. So if we hear before this call is over, then I'll let everybody know.
Mm-hmm.
It's actually November the 1st that we tried to reschedule at the same time. So let's hopefully maybe we'll hear from him before we go.
And
Anybody
Jeff,
else? I mean, you want to stay with? Are there other
Jeff,
hands up, Michael?
Yeah, there's Jeff. We have Jeff. Thanks for being patient, Jeff. You want to ask your question or bring up your comment?
Yeah, no worries. I was going to mention Matt's case specifically. Matt Sharp, who is, we're not telling tales out of school. He shared this publicly at many meetings.
Yes.
But he was in one of the Sangamo trials. And to my knowledge, he wasn't followed at all. And that was in the Sangamo trial. I would imagine because, UCLA, the trial they did there, they've got probably more stringent follow-up requirements. But correct me if I'm wrong, anyone, but Matt has said that they didn't follow up at all, to my knowledge, once the study was over. And in fact, I just
think
messaged
Matt,
him to see if they have.
I think the two Matts,
Yeah.
One was as Jeff indicated, he was much better for a really, really long time, and I think
Yeah.
remains better. The other is off therapy completely. He's Jeff too, the other Jeff, but they were both with the
Matt,
same
the other
doctor,
Matt.
who I think once the money stopped, he
Yeah.
was out of there. Right. Unfortunately, that's I think what happened. I can't explain it in any other way. They both try to. I know one of the Jeffs tried to get in touch with him a number of times. I mean, I'm
Matt.
sorry, Matt.
Matt.
Well, usually
I
there's
am
so
not
many
cured.
Jeffs, right? I'm sorry, everybody. So, I really, we can't, there's no way for us to figure out what happened there. But interestingly, Alster, who was at UCLA, did get followed. So, there it is.
Well, I think that's that might have been an institutional requirement as well as
Yeah.
You're right.
Yeah. I don't know if you can see my screen, but the long-term follow-up study is shown there, but notice that it didn't start or wasn't posted until December 2019, which was a long time after those trials ended.
Uh-huh.
yeah.
So
Do
I
we
think-
know if it was ongoing before that or
Press
did they?
releases.
Oh, okay.
And then the other
Sorry,
thing I
I
wanted
need
to
to-
mention is to kind of follow up to what Linda was saying. Maybe we should have a call, maybe one of these calls with the FDA and kind of get their thoughts on.
Well,
Yeah,
Jeff,
how-
I tried. I don't know if you
Did you?
heard me say that, but I don't know. It's been a number of months now that I got in touch with them. Oh, there you go. Got it. And asked them, again, I'll say that every year we do a meeting with CEDAR. We did before
Yeah.
COVID, and then, the drug side of the FDA. And I wanted to start, having that same thing happen with CBER, the biologics side, where most of the cure trials are taken, reviewed and authorized, whatever. Anyway, the lady was really gung ho at first. Interestingly, instead of me getting a doctor, I got somebody from their PR office, for lack of a better word, communications. I don't think it's PR in the usual sense, but anyway, she was, oh yeah, well, let's, I'll be glad to help you and yada, yada. And I kept getting back to her, but in the end, she hasn't gotten back to me. So I have other people there I can contact, but again, I'll try and do it again before the year ends, and we can maybe hopefully have something with the FDA, afterward next year. Because really, all we have is questions about this kind of stuff. So, I won't give up, but it's not been easy, and it's COVID, so everybody's busy. They're all crazed with vaccines, and that's a different department. But so, do we want to stay with safety? Oh, I'm sorry.
Well, I just wanted to follow up on that. So, do we want to have maybe a smaller one-off meeting as opposed to a more public meeting like this, where they could be a little more forthcoming?
Well, that was what I was
Radar.
gonna do. It wasn't a
Yeah.
public thing. It was just
Okay.
gonna be, how we usually do with Cedar. I mean, we used to do it at Croy and oftentimes, I mean, in the end, it got to be pretty large, but it doesn't have to be, and it could be
Mhm.
whatever
Mhm.
they want, but they need to get back to me. So,
Yeah,
Right.
okay,
but I'll keep on it.
thanks. We know
Do
you
we
will.
want to stay with safety? Or other people have other issues we want to discuss? Richard, do you want to keep going? I'll
Well,
see another slide here.
I mean,
yeah, go ahead.
Yeah, you
I was
go,
just going to
Richard.
say, I can give a quick recap for the people that have joined. So
Yeah, that's
because
great.
Dr. Halber contacted us last night and couldn't do the presentation today, I just shared a couple of bits of news that have come out in recent months
Mhm.
about some HIV gene therapy trials. One of them being this first trial of an approach, gene editing approach to target the HIV provirus from Excision Biotherapeutics. This trial here is single dose that you asked about, Michael.
Mhm.
And the first person has been reported to have been dosed, has been followed for a few months without any safety issues arising yet. And then the other bit of news is that American Gene Technologies, which has this strategy to try and genetically protect the CD4 T cells that are designed to fight HIV, has initiated this ATI portion of their study. So we should get some news by the end of the year about whether this kind of targeted approach can allow the immune system to better control
Right.
viral load or not.
And
And the
what
other
was-
thing,
I was going to just point out, also there's the anti-HIV duo CAR T cell trial. They've also dosed their first participant. That's, just another update that Karine put into the chat. I don't know if Karine, you know anything more, or you want to talk a little bit more about that?
No, it's just really exciting that the dual CAR study is up and going. I don't think they'll be dosing a second participant until maybe October to November. And we're also working with the animation lab on a video about dual CAR. We just need to get it re-recorded. We can share an older version with this group. It's a really exciting technology and we're very hopeful. And there might also be another similar study being planned in Uganda as well as a sister trial.
And the diva of, which is not a diva at all, but Kim Anthony Gonda will be with us next month to talk about that exact topic, the duo CAR T cell approach. So we'll have the whole hour on that, and I know she'll be here.
Right.
So stay tuned for that.
We just saw someone saying that they dosed the second patient yesterday at UC Davis.
Oh, cool.
Awesome.
Awesome.
Yeah, that was interesting. In that study, I mean, it started out at UCSF, and we couldn't get any patients there because there's like for 30 days you have to be accompanied by a responsible adult 24 hours a day. So unless you live with somebody, it's kind of hard for that to happen. Even if you live with somebody, it's kind of hard for that to happen. And I think the problem was they had neurological toxicity in cancer patients who were much more sick. And, as always, we hope that people who aren't that sick with HIV, those sort of toxicities won't happen, which has pretty much been the case in other arenas. But anyhow, it was easier to get people that could meet that requirement at UC Davis.
Mm.
It was a long time setting it up, so it was just a nightmare trying to, go from one place to another and all that. But Kim can tell us about that all next month.
Yeah.
I'm sorry, Richard, go ahead.
I know that's all I had. I mean, I'm sorry, apologies to everybody that we had to kind of busk it today. Hopefully
Yeah.
we can get Dr.
Well, let's talk
Alpha
about
with
what
us.
other people are interested in as far as this is safety. What, what other questions people have? I mean, if we did have an FDA meeting, what would you like to know from them? It would be helpful for me to know that.
I mean, I would love to know exactly what their plans are if a company falls back and is disbanded, and who's following, who takes over the following of participants, or is that something that's just left to scatter?
Right. So let's start with the long-term safety,
Right.
what that looks like.
Right.
And how that is amended,
Mhm.
you know, what is it 15 years, 20 years? I think it's 15, I don't know, but you know, and then what do we do if a company goes out of the business? And, you know, I just think it was not about the company per se, because, you know, it's not like they disappeared. They're still out there. They just decided that there
Good.
wasn't enough money in HIV.
Right.
So, I mean, but it's still a good question. I mean, I think we
Yeah.
need to know all of this stuff.
Michael Luciano, I see you have your hand up.
Hi. I'm trying to think how to formulate my question exactly.
Okay.
I know that many of these approaches are still in very early and or mostly theoretical stages at this point, but I wonder what efforts are being made to include and to address people with complicated treatment histories and or with comorbidities due to aging, especially since, as we are constantly reminded, 50% of people in the US living with HIV are now 50 years of age or older or will be momentarily. So, many of the studies I have heard of, at least, are recruiting people who either are very early in the course of infection, attempting to cobble together or attempting to hit HIV extremely early in infection in order to limit the size of the reservoir, and and other approaches all specifically exclude people with a long list of conditions or with a history of treatment failures, etc. So, is a cure that comes out of this going to genuinely be universal, or is it going to be restricted to a very small number of people who are at the appropriate moment in the course of their HIV infection? And I've seen very little addressing that.
I think the problem with this is, and believe me, there's a group of us who are, for a better word, always screaming at companies about immunologic non-responders, people who seem to be different, whatever. But the problem with the inclusion exclusion criteria that you have described at this stage is not the trial itself. It's the treatment interruption. Okay, so when you have people with lots of issues, with that have been chronically infected for long periods of time, that have all those comorbidities, they're in much more danger of bad outcomes or worse outcomes than people who don't have the comorbidities. So it's about the treatment interruption that's where all of that stems from. So what we need is something to measure the latent reservoir. And I mean, everybody's working on that, but that's the reason for it. That's the reason for the age, exclusion, and that's the reason for all of the other conditions being listed.
Mm-hmm. Jeff, did you want to bring up your follow-up question?
Yeah, no, thank you for raising that, Michael. As Linda says, that's something we are always yelling at them about. But I think, and then the point we're trying to make, and I think we need to be doing this across the board in the ACTG, the Delaney collaboratories, every place where this is happening, at the university cabs, is that it needs to be scientific criteria and not just an arbitrary age limit. They need to define what those concerns are. And the comorbidities are one, and that's a perfectly valid thing. But there are also plenty of people, in their 60s and 70s who are doing just fine, who don't have, really bad aging comorbidities, who would be good candidates for these. So that's something, we're always advocating for. But I think maybe one way to address that is for the community to come up. And again, we can do it in the context of the ACTG and MDC, but I think we need an overarching research agenda priority for HIV and aging that raises these concerns, and list things like that. Follow up for gene therapy trials, aging restrictions. I think all these things that are concerned, we need to put down in a document and say this is what the community wants. We can hand it to researchers and say, and the FDA for that matter.
Yeah.
So
Well, the API part of it is scientific, all but that age number.
Exactly. That's my
The
point.
conditions are all scientifically based, but that age number, and we've gone back and forth with them on this. And we were, if you remember, Jeff, we were at 65 instead of 60, but then COVID came.
Right.
And so there were dangers of taking older people off of meds. It's so complicated. But I agree with what you're saying about the age number.
We just need to keep pushing and not let them
Yeah.
get away with these
Yeah.
arbitrary age restrictions.
Alistair, what did you want to bring up?
Actually, if I can follow up on what we've just been discussing.
Sure.
In terms of a lot of studies, the age 65 doesn't necessarily make as much sense. And if I remember correctly, for the Anchor study, initially they are, maybe from the start, they had something in place that when you entered the study, according to a particular measurement tool, your life expectancy had to last at least as long as the end of the follow-up of the study. I don't remember the name of the particular tool, but there was something that you put into several factors, including your own age, whether or not you'd high blood pressure, a couple of other things. And
I think it's the Karnofsky score.
Sorry?
I think it's called the Karnofsky score.
Okay. But basically, in a study for the follow-up, maybe 15 or 20 years, if the life expectancy of the participant is less than that, according to that score, then does it not make sense to not allow that participant into the study?
Well, now
And
you're
if
talking
it's a
about
20-year follow-up,
a way different kind of study, though, Alistair, than these
right.
first in human studies.
Right. The other thing is that when the study is only going to have 5 or 7 participants, I'm not sure how reasonable it is to start off with people who may not actually survive to the end of the follow-up period. Just on a statistical basis, it would be ideal if a study could be started and could enroll a couple of thousand people of all the various criteria and then follow through on that. But that isn't the practicality for some of these studies where it's very early stages and whether or not it's likely to even be effective.
Yeah, and I think it goes back to the original question about the sort of, that so far these kinds of gene therapies are only being studied in fairly restricted populations. It's just, we haven't unfortunately reached the stage where there's enough of an efficacy signal from any of these studies to justify what would usually happen, and what we've seen happen with antiretroviral drugs, et cetera, which is, you slowly expand the population and loosen the criteria for participating in the studies as there's a reason to move the intervention into efficacy testing. And I think if it was possible to get something that was going to move into a larger efficacy study, then those kinds of restrictions would be a lot less.
Right. The question I was wanting to ask that was completely different is, back in the time of the Sangamo study, I had to seriously think whether or not I wanted to enter the study, because one of the things that was mentioned in it was that participating in this earliest trial would make me ineligible for any future similar trials. And that, to me, I had to seriously think, okay, do I want to sort of roll the dice this early in the game? Or does it make more sense for me to actually hold off and let somebody else do that? And I'd be able to take advantage of the potentially more likely to succeed studies later on. And I decided that since nobody else was doing it anyway, I might as well just get in on the ground floor and see what happened. And as a result, is each new study of gene-modified autologous stem cells came out, I had to mutter and grumble a minor amount to myself because I didn't think I was eligible. Then the past couple of years in presentations, I've been hearing about people that were in their third study of the same type.
Yep.
So has that particular criterion been lifted?
I think it's study by study, you know.
I was going to say, I think it's something that we're used to from the prevention realm. In other words, if you've been given a vaccine candidate, they don't usually let you into another vaccine study, but until we start getting efficacy signals with gene therapy, at least I know this is from the University of Pennsylvania, Jim Riley was the one who presented it. They allowed that person to go into 2 other trials after the first trial. And it seems like him having these 3 different interventions has led to some sort of form of control. So they're just looking at it as, you know, catch as catch can, and they're going to follow him anyway. So they're going to try to unpack what in each of those therapies may have led to him controlling HIV finally. And so I think it's just a little bit of case by case. There's no standard. In other words, there's nothing that says that you couldn't have another gene therapy intervention after your first one. I think it's just that's the approach that we have based on the HIV prevention trials. The idea that once you have one therapy like that, therapeutic intervention, you don't want to have another, so they can separate which one was the one that was efficacious.
You know, and that person might be excluded from like a phase 3 trial later on.
Right.
You know?
And I can just flag here that there was a more recent stem cell modification trial at City of Hope where that was an exclusion.
Oh, was?
And I think for the reason that it, you know, they just not wanting to have the data analysis complicated by the possibility of something else contributing.
I mean, I always alert people and warn people of that when I do my little talks. I mean, that's something that you should always look for and consider.
Yeah. There was also an interesting question that James posted, and I don't know if anyone wanted to wax on it. He was asking for people's opinions on which approach do we think is the most hopeful one so far in terms of the interventions that are being tested. I don't know if anyone wanted to begin. I know that there's no signals yet that we can say, oh, well, this one is showing something.
Well, you know, just from, you know, intuitive sort of whatever. I mean, to me, the CRISPR seems like it's more dangerous, although we heard Pretty last month talk about how she's doing things that actually don't do you know, it's more of a bolt in or whatever, as opposed to a cut.
Right.
So that makes that less dangerous. And I think we all know that as the studies proceed, things get better in a lot of those regards. Not always, but often. But to me, it seems that, you know, I like the idea of the duo CAR T cells because I think, you know, the main point that I have learned in HIV is that you need combinations, you know, and so why wouldn't you need a combination here as well?
Right.
I mean, you know, we have these kitchen sink studies where they're not, especially for chronically infected people, where they're not only doing an intervention, but they're also pumping up the immune system with other things because, you know, I mean, some people, and not everybody's not equal in the state of their immune system for this.
Right.
So to me, I mean, I don't think we're going to see a magic bullet in HIV. I really don't. It's too complicated and it's kind of like aging. It's so many different things involved.
Right.
So, but anything with a combination approach seems like it makes me take a second look.
I think it's a really tough question, but I think by the time that the conference on retroviruses and opportunistic infections rolls around early next year, that there will be some more data from these studies that will maybe help show, you know, which might be the most promising.
And Richard, we have a request for someone asking for the TAG website address for the trials so they can look at it themselves.
And apparently Alistair's having trouble with your links or something. You need to take a look in the chat. He's talking about a link that didn't work or something.
It wasn't that the link didn't work. It's that when screen is shared, the links that are on the shared screen are not clickable.
Oh, I see.
Right,
But
right.
the person who is actually sharing the screen can cut and paste, just as Dr. Jefferys did,
Yeah.
to the clickable link, and it makes it much easier to follow along at the same time.
Yes.
Thank you.
Sure thing. Sorry. Yeah.
Cool.
Ooh.
Okay.
Hi, it's
He's
Mark.
not
Milano.
Dr.
Can I
Jefferys.
friend
I
it?
apologize.
Go ahead, Mark.
Go, Mark.
I'll just go with my gut too, that I'm much more excited about approaches that are looking for a functional cure rather than a sterilizing cure. I agree with you, Linda. I'm 66. I'm quite sure I'm not going to see a sterilizing cure in my lifetime. It's just too difficult to get every scrap of virus out of the body. But the approaches that want to retrain the immune system to handle HIV the way the immune system handles chickenpox, that I think is very doable. So those approaches, I think are much more worth pursuing. And I think as advocates, we should be pushing more for those, because those are going to be achievable in our lifetimes. The total eradication, that is decades away, and people aren't going to live to see that. So I think as an activist, I want to prioritize what is doable in at least a reasonable time frame, and that's a functional cure. Do you agree, Linda?
Oh, yeah. And I think that's what's happening. I just don't know they'll ever get to that point unless your own body does it like Lorraine, it's
Right.
It's just not going to happen.
Right.
Right. Yeah, but I agree with you as well. I think it's interesting to talk to scientists about, hey, let's stop making such a big deal about these bone marrow stem cell transplant recipients, because that's going to be a small pocket of individuals who will ever access that. I mean, not that we shouldn't learn from those, but what about the long-term control of HIV for 15 years that was just right besides the announcement of the 5th person to be cured by a bone marrow stem cell transplant? You would think that IAS would make more of a big deal about this person who's been off medicine for 15 years, because that is not only more achievable for a wider group of people, but that is the next best thing to being cured, isn't it? To not have to take medicines, to not have the side effects, to not have to worry about the costs. I think that's really where things are lying. And it was interesting talking to scientists, getting them to understand that it's not so much that we don't like the bone marrow stem cell transplant recipients or that news, but to make such a big deal over those cases that are going to be limited and their effect when there's other cases that we could be talking about a lot more, the post-treatment control.
And control.
We've been after them about that exact thing. Why do some people not get sick, or not even get it, period. But it's age old. I mean, we started that with the NCI when Martin Delaney was alive, before we even were discussing cures. It was really just like, how do we, I don't know why they don't. I mean, Reagan has been doing this for years and years, though. They've been working on it for years.
I'll tell you why they do it. Why they do it is because it's press. Curing somebody is press.
Yeah.
Putting somebody in remission for like 5 years and then having to redo the treatment, which would be great, right? Treatment once every 5 years and remission in between. That'd be fantastic. But that's not exciting, For the media, a complete cure is exciting, and that's why they promote that stuff. They need to promote what is actually useful, which is long-term remission. That's what's useful to people.
Yeah, I agree with that.
Although it wasn't gene therapy, there was a bit of good news that was published yesterday. This study with a broadly neutralizing antibody and a latency reversing agent that were given at the time that people in the study were starting antiretroviral therapy, and they did see a significant acceleration of clearance of the reservoir, and they, I think, had 1 or 2 examples of people who displayed a lot of sort of good control of viral load after a treatment interruption. So that was just published in Nature Medicine yesterday. And I also need to just issue a disclaimer that I am not a doctor in any way, shape, or form.
I was waiting for that. But he is our Encyclopedia Britannica, so that's a doctor.
Not a doctor, but he plays one in ATEC.
He's smarter than most doctors I know.
I don't think that's true, unfortunately.
Well, more informed, maybe.
Yeah, there you go. There you go.
Yeah.
But
But what else are we interested in?
I think maybe we should give people their time back.
Yeah. It's up to you. If there's no other
Well,
questions.
there's about 30 people left. What, do you want us to keep talking? You want to keep discussing things, or do you want your half an hour back?
I think it's actually 10 minutes.
Yeah.
Oh, really?
Yeah, we've
Oh, well,
we've
let's
filled
just
up most
wrap
of
it
the
up.
hour.
Yes, it was the last time I looked, it was 12:30, so.
time flies when you're having fun.
There
Right,
you
I
go.
well, time
appreciate
flies
it.
whether you're having fun or not, is what I always say.
Been a
This
great
has been a very
discussion.
interesting discussion, and I thank you for managing to shift into it.
Yes, thank you. And thank you for sharing what
Told
you
you,
did, Alistair.
Encyclopedia Britannica. Well, please join us next month for the duo CAR T cell discussion, which is really extremely interesting. And
And
if we hear back from Dr. Halber, we'll be in touch about 11/1.
yeah, and so we're going to try to reschedule it. And just so you know, since you are registered for this webinar series, when we do change it, you will automatically get the update. So you won't have to worry about like a special email coming through. It'll be updated, and you should get one of those emails sent from Zoom once we do schedule that. So we're looking at November 1st, but don't quote us on that. We need Dr. Halbert to actually confirm with us first before we go ahead and schedule it. So I just wanted to let you all
Real
know.
quickly to see if he's gotten back to me yet for anybody.
And Dr. Halber has a kind of interesting variation on the approach of the sort of genetic surgery of excising, cutting out HIV from infected cells that doesn't
Right.
use CRISPR. It uses a different kind of technology.
He also is working with Hope, which is one of the MDC collaboratories.
Mm.
I don't see anything from him yet,
Okay.
so.
Well, we'll let
And
everybody-
Michael,
Yes.
Michael, even though Dr. Harper wasn't here, I came in late, so I love the video and all the links, even the links in the chat. I know that's a hassle, but if we could get those so I can click on those, I'd love that.
Certainly.
Thanks.
You're welcome.
Okay.
All right.
All right, everybody. Thank you for hanging out and staying
Yeah.
with us, and we'll see you next month.
Yes.
Bye,
Awesome.
everyone.
guys.
Thanks,
Thank you
Michael.
all.
Again, everyone.
Bye.
Happy,
Bye for
happy
now.
Halloween,
Richard, for
everyone.
stepping
Take
up.
care. Yes. Happy Halloween.
Bye-bye.
All right.
Michael, you got time for me to call you now for a minute?
Yes,
real quick?
yes, yes.
Okay, I'll call you right back.
Okay, great. I'm going to sign out.